welcome to Layla Teachers. Today's topic is enzymes, and it's a huge topic, so we'll split it. This is the first part. So first, let's define enzymes. What are enzymes? They are biological or protein catalysts that increase the rate of reactions without being changed in the overall process. So here's a reaction that makes it clear. An enzyme reacts with substances called substrates, and then they form products. Now, the enzymes are not a part of the products, they are separate from them, but they join the substrate, allow the catalytic reaction to happen, and then form the product and then leave the product. Okay, moving on to the properties, but before that, I'll just tell you that RNAs, they have catalytic activity, but they're not called enzymes, they're called ribozymes. Now we will move on to the properties. So the first one is active site. So enzyme molecules, they contain a special pocket or a cleft, some kind of space called the active site, and this is where the substrate fits. Then there is something called catalytic efficiency, which is like the rate. So the number of molecules of the substrate converted to the product per enzyme molecule per second. It is called turnover number. Second, we'll speak about specificity. So there are two types. There is reaction and there is substrate specificity. Reaction is where one enzyme is specific to only one kind of reaction, for example, only decarboxylation or only deamination. And substrate specificity is where one enzyme is specific to only one kind of substrate, for example, only glucose, not fructose, not galactose. All right. Some enzymes that require non-protein components, and this is the holoenzymes. If it is a metal, it is called a cofactor. If it is a small organic molecule, then it is called a coenzyme. If the coenzyme is permanently associated with the enzyme, then it is called a prosthetic group. FAD is an example. And the last property is regulation. So enzymes can be either increased or decreased. Okay, now we'll move on to nomenclature. So enzymes have the suffix ase. For example, lactate dehydrogenase or urease or glucokinase. But some retain their original names, like trypsin and pepsin, which are in our bodies. And each enzyme is assigned a classification number, which you do not need to know. It's just for your information. Now we'll move on to the classification. So there are 6 major classes with numerous subgroups, but we'll only speak about the major ones. So first is oxidoreductases, and what do they do? From the name, you can say that they catalyze the oxidation and reduction or redox reactions. Next is transferases, so they transfer carbon, nitrogen, or phosphorus containing groups, for example, serine to glycine. They're amino acids. Then you have hydrolases. They are responsible for the cleavage of bonds by addition of water. Then you have lyases. They're responsible for the cleavage of carbon-carbon, carbon-sulfur, and certain carbon-nitrogen bonds. Then isomerases, which are from the name, you can say they're responsible for the racinization of optical or geometric isomers, so cis to trans or vice versa. And ligases. Now, these are responsible for the formation of bonds between carbon and maybe oxygen, sulfur, and nitrogen, but they're coupled to the hydrolysis of high-energy phosphates, so they are energy-requiring reactions. If you want a mnemonic to help you remember the classification, here's one. Only the happiest lady is laughing. Okay, now the next part of the video is about isozymes. What are isozymes? They're also known as isoenzymes, by the way. So each of 2 or more enzymes with identical function but different structures. For example, glucokinase, it is a variant of hexokinase. And then we have another one, CPK, which is creatine phosphokinase. It exists in 3 different isozymes. One is the subunits are BB for the brain, MB for the heart, and then MM for the skeletal muscle. Okay, moving on to the main part, the mechanism of action. So what do catalysts do? They provide, enzymes, they provide an alternate, energetically favorable reaction pathway, which is different from the uncatalyzed reaction with regards to the free energy of activation. I will show you in a little while a small graph to make it easier for you to understand. So you can see that there is the enzyme with the substrate. It forms the enzyme substrate complex. Then you have the catalytic step, which is the enzyme letting go of the product and completing the reaction. So the S and the ES, they are the substrate binding, and the ES is known as the transition state. It is the transition state stabilization. Then you have something called the lock and key model. It is a theory where the enzyme acts as a lock and then the substrate acts as the key. So they fit in like a lock and key. Then the next theory was induced fit model, where the enzyme conforms its shape to fit the substrate. So the induced fit theory is more looked at these days. Here you can see that with no catalyst, the activation energy looks higher than with an enzyme. So this is why enzymes provide an alternate route for the reaction. I hope you enjoyed this video. See you in my next one. Bye.